Hi, good evening. Hello. Am I audible and visible? I mean, is my PPT visible? And someone of you put it in the chat box so you can unmute your mic and say. Okay, thank you. Okay, so good evening, everyone. This is the first class. This will be the first class for this course, Plant Modeling. My name is Shruti, and I'm a PhD scholar in Indian Institute of Technology, Palakkad. And I'm taking this course as a part of my teaching assistantship for the PMRF scholarship that is and the Institute of Smart Structures and System is the platform that is helping me to conducting these classes. Um, so before jumping into the course, these will be the detailed syllabus of the course. I think I've mentioned the module names in the flyer. And this is just the details of what we'll be going through in each of the module. And I'll also be sharing the syllabus with you either in the WhatsApp group or I'll be mailing it to you. So please make sure that you've joined the WhatsApp group because probably I'll be using that much more in frequency for the communications rather than the emails. And I hope all of you are okay with that. If someone has any issue connecting with the WhatsApp group, please let me know in the chat or yeah by email. So this is the detailed syllabus. Today we'll be going through the an introductory part of this course, which includes what is plant. I mean, this is mostly for all those who are not coming from a botany or biology background. Myself, even I'm not a botanist. My specialization is in civil engineering. So this course will be more on a perspective of how someone who's not very familiar with the leaf and botany can understand the concepts that is required to model plants. So when it comes to plant modeling, we might not be able to go in depth into a lot of plant processes and terminologies that is related to the plants. We will only be going through those aspects that is required for us for the purpose of plant modeling. So before I go into this course, one tool that I'll be using side by side with the theory is a crop model called the BioCro. BioCro is run using an R script, and these are some of the links you all can go through to familiarize with the model. So, due to time restrictions, I mean, I will be only taking like 25 hours of this class, so I will not have the facility to teach the entire software. But there is ample resources online in their website and in their GitHub page where you can go through the model, you can read about the model, and here, if you see, these are the research papers that you can go through to understand the model in terms of its application. And these are all the documentation, the model, the codes, and everything about the model. So, like I said, I won't be going in too much detail of how to run an R script to use this BioCrew model, but then I'll be sharing the codes with you So you also have to take it a bit of a time to understand R script, and which will be helpful in running this biocro model. Like I said, it's about modeling, so I think it is necessary that you enhance your coding a bit of have some knowledge about the coding part of it, or running using an R program. So I won't be covering the R language or application of R language for BioCro. I'll be presenting it to you, explaining how I'm using this model that is incorporating all the theory that we are going to be studying in simulating a plant or simulating the processes in plant. So this is the detail about the model that is going to be used in not in today's session, but in the upcoming session. So I'm giving this in advance so that you can have an idea about all of it. Moreover, I have also shared a Google form where you can post in your doubts, and I will try to attend to it to the maximum. And also, during the session, I would appreciate it if we can keep the doubt sessions towards the end of the classes, because I'll be recording these lectures. So it would be helpful for people who are viewing the videos to have an uninterrupted lecture series, and we discuss the doubts towards the end. Please feel free to drop in your doubts or points of discussion in the chat. That also I'll look at during the end of the session. So today's lecture is an introductory lecture. Uh, we will be just talking about what is this plant ecophysiology and some uh, plant portions that we require for the modeling purposes. And I believe a lot of you over here are um, people with a good background of botany. So you can also, point out if I'm saying some blunders, because, like I said, my specialization is not botany, but I'm covering these portions which are only required in the aspect of plant modeling. So all of us know about plants, how important plant is, how complex plant is. Plants are organisms that, unlike humans or animals, cannot go to different places to uh, get food for their growth. They are organisms, or they are living beings that make use of it. They manufacture their own food with the help of carbon dioxide, light, and water. And so they are called autotrophic organisms. And other heterotrophic organisms like humans and other animals, we feed on other animals or plants for our food, while plants are uh, living beings that have the capability of manufacturing their own food. So a plant, although I believe many of us, I don't know how many, but a few of us over here would have stopped learning about biology after our class 10. And our knowledge of plant is probably just stuck to the fact that, yeah, plants are manufacturing food, glucose, and releasing oxygen by taking in carbon dioxide and light and water. But plant is a very, very complex system which is capable of surviving on its own. And there are different branches of plant science, sciences that study about its cell, sciences that studied about the genetic makeup of plants, sciences that the plant science branches that study about the uh, chemical reactions that are happening in plants, the timing of the germination of seeds, the flowering, the dying of plants, and much, much more. So our focus and major focus is the interaction of plant with the environment, and also the basic physiological functions of plant. So our plant modeling class will be focusing a lot, or will be going in depth into plant physiology and plant ecology. Plant physiology is nothing but the branch of plant science that is dealing with different life functions of plant, and plant ecology is the study of how plant function in an environment and interact with the environment and its surroundings. And we'll also be going a little bit into the chemistry of plants, but then it's we'll just be touching about it. We won't be going in deep. But for all those processes that are involved in plant physiology, what are some of the chemistry related to that? So basically, plant ecophysiology is nothing but a combination of the study of plant physiology and plant ecology. Ecology, like I mentioned, is the branch of science that is studying the relationship between plant and their interaction with each other as well as with the environment. And plant physiology is the study of plants, focusing on how they are functioning, particularly an individual plant. So when we club these two sciences together, we have the plant ecophysiology. Plant ecophysiology has also various other names. You can know about it in physiological plant ecology, plant physiological ecology, and plant autoecology. So, like I said, physiology is one major focus of our class. So, there are various physiological processes. One key process being photosynthesis. Another is the respiration. Even plants, they not only take up carbon dioxide, they release oxygen also as part of photosynthesis, as well as part of respiring for their survival. Then there are processes that deal with the nutrition of plants, plant hormone functions, tropism, which is the movement, directional movement of plants to various stimuli, nastic movements, which are the non-directional movements of plants to stimuli. Circadian rhythms are the ones which are actually giving the plant an idea about what time of the day it is. Then there are physiological processes that define the stress conditions, experienced by plant, germination, about the inactive state of plant, stomata function, and transpiration. So, in out of these, the first two, photosynthesis, respiration, stomata function, and transpiration, are the most important key physiological processes that we will be focusing on. And this model that I just talked about, BioCro, they have a very efficient method to simulate all this process or predict all these processes if we give certain inputs that are required. And plant physiology again is not just a separate part of branch of plant science. It is interacting with all different parts, plant sciences like morphology, ecology, phytochemistry, genetics of plants, and everything over there. So, like I said, our focus is going to be photosynthesis, transpiration, and stomata function. Now, why should we all study it? I mean, from the perspective of different backgrounds. I think from the Google form, I did get a few of them into environmental sciences, into agricultural biotechnology. There was a mathematics student. There are other branches of all sorts of branches that are coming in for this course. So, in the perspective of multi subjects, or why this subject could be useful to a lot of people, is because one is some input we can give into agriculture. Now, we are all living in an era where climate change is having a huge impact on us because of various processes like global warming, and food security is being an issue. So, in order to develop a sustainable agricultural practices, the information about plants, I mean, going in depth into plants is a very necessary factor. Similarly, when we involve in acts of conservation, even for plants as well as in conservation of other species, it is necessary that we know how plants, which is an integral part of the ecosystem, um, how does it function, which can help in enhancing the acts of conservation or methods of conservation. Then again, climate change. I think plants is a way, the natural solutions using afforestations and plantation agriculture and different sorts of sustainable agriculture is a very recognized method for mitigating climate change. So, in that factor, also, plant is a very important dynamic component that we have to go in detail to. Again, preserving forests requires knowledge about the trees and plants, and plants are also now as replacement to fossil fuels, biodiesels, bioethanol, biofuels can be used. And also for this purposes also, it is necessary that we have a basic understanding of how plants function, apart from just using plants as just one single entity named plant in and including it in all of these studies. It is necessary we go a little more deeper into plant the physiology of plants. Now, this is also just giving a basic idea about how the study can be made use to different arenas and areas. Uh, like electrical, there are departments or there are centers over here where electrical engineers are involved in developing sensors along with IoTs to sense the health of soil and in turn help in using that information for sustainable farming. Then genetic engineers and biological engineers can incorporate this knowledge into developing genetically modified crops that can, mitigate this issues of food security, improve crop productivity. Then again, computer programmers can use this knowledge in developing models, strong plant models that can represent it in a more accurate way. Again, climate consultants, I think the knowledge of crops is very necessary to incorporate them into climate change studies. And all of the fields, I think now it requires all of us to follow an integrated approach rather than a very singular approach to find solutions to a lot of problems, especially in terms of in the climate change studies. So plant knowledge is useful in a lot of ways, and I think everybody it is necessary that we have a basic understanding of how these plants work, and we will be doing this course just to step into that discovering what are the basic processes that are actually very important in plants that can contribute to different fields of study. No, this is one representation how plants are important in climate change mitigation. So what you see here is a global carbon cycle that was published some in a few years ago. So according to this, you can see that there are these blue color. That's my cursor. Okay, so you see blue color over here. The blue the things that are written in blue are components of the ecosystem or the terrestrial system that is sorry, not terrestrial, of the system that is actually storing carbon. So atmosphere is storing 750 pentagrams of carbon. Fossil fuels are storing around 5 to 10,000 pentagrams of carbon in within the earth. Earth's crust itself has got a lot of carbon. Soils are having this much of carbon. Plants also, the biomass of plants, basically the stem, the leaves, roots, all are composed of carbon only. So they have 560 pentagrams of carbon stored in them when you look at the entire Earth as a unit. Oceans are also carrying a lot of carbon. As it goes deeper, the carbon storage is higher. Now, what happens is So we see here that there are so many red lines, arrows that are going outwards and also going inwards. So all the arrows that are going inwards is showing how much of carbon is taken from the atmosphere into uh, wherever the arrow is directing. So if you see here, photosynthesis is taking around 120 pentagrams of carbon every year. Uh, into the plant body. So it is storing it in plants. At the same time, here volcanoes are releasing zero point 0.1 pentagrams of carbon every year into the atmosphere. You see, deforestation is contributing 1.1 pentagrams of carbon into the atmosphere, and you see a lot of this one here. Ocean is taking in so much of carbon as well as releasing carbon. So these are the atmospheric fluxes that is being shown in red. Not just atmospheric, but fluxes between every the earth and the atmosphere, water and atmosphere, land and water, all of that. So you see here, if you look at this number over here, I have put the numbers, which is the carbon sinks, that is the carbon that is taken from the atmosphere away from the atmosphere as green color, and we see ocean and photosynthesis are the two processes that is contributing to carbon sinks or reducing carbon from the atmosphere. So this is again pointing out to the importance of the process of photosynthesis in taking away the carbon dioxide from the atmosphere back into the soil. And these are some studies that there are several research works and studies for those of you interested in research and studying more about how photosynthesis is very important process when it comes to acting as a carbon sink that will help in reducing the carbon dioxide concentration in the atmosphere. Then you can go in deep into the photosynthesis processes which will help in improving crop efficiencies, increasing the efficiency of photosynthesis in plants is in turn going to help in a lot of benefits to the plant by helping them in making increased produced and also making effective utilization of the water that is provided to them. So there are several areas to which we can divert this knowledge in terms of research also. So as we'll be now moving on to those parts of this plant physiology or plant structure that we require for our course, and we'll start with how plants evolved from non-vascular plants to vascular plants. So non-vascular plants were the first kinds of plants that appeared on Earth. And non-vascular plants are plants that do not have a very complex transport system for transporting water. And it is from this that vascular plants evolved. Vascular plants are plants that have a very complex and specialized structure for transporting water and nutrients. Their structure is also more defined. They have stems. From stems there are branches that that has leaves. They have a root system that's fixing the structure vertically on the earth. So theirs is a more complex structure. So we will be going into processes that is involved in vascular plants. So let's scrutinize our focus onto a single plant. So this is the structure of a typical plant. There are different types of plants, different types of structures, but we will just be discussing a typical plant. In a typical plant, there are leaves, stem, roots, and the plant will have flowers. From flowers, there'll be fruit, and so on. So stem is the one that is, let's start with root. Roots are the systems in a plant, structure in the plant that holds the plant towards the soil. They not only hold it, they also play a huge role in absorbing nutrients and water from the soil that is necessary for the growth of plant as well as for various processes of plant. And also they store a lot of sugars. There are different types of plants where the tuber crops and all which store a lot of carbohydrates in their roots which are edible to us. When we move on, stems is the is the structure or the plant part that is providing a structure to the plant. It is playing a huge role in not only providing a support to the plant system, like a backbone to the plant system, but plays a huge role in transporting, helping in the transport of nutrients and water. Then we come to leaves. Leaves are the a very important part of the plant, which is forming the basis for the food production of plants. So it is in leaves that this very important process called photosynthesis occurs, and not only photosynthesis, which is the release of oxygen and intake of carbon dioxide, but there's another process called the transpiration that happens through plants, which is the removal of water from the surface of the leaves back into the atmosphere. So that is called transpiration. Transpiration is the release of water from the surface of the leaf to back to the atmosphere. So from all of these, we know that photosynthesis is happening in leaf. So now let's go more deeper into leaf. We won't be focusing a lot on the stem and roots, but we'll be going uh, in a little more deeper to the leaf structure. So leaf also, it is a very complex system. You can see, as you can see in the figure, it has got a leaf blade, which is basically the entire flat part of a leaf. It is attached to the stem through a petiole, and the base of the petiole you find the axil, and then it has got a lot of veins through which water is transported. So our focus will not be on any of the stem or petiole part of it, but it will be on this leaf blade, which is the flat part of the leaf, typically green in color. This leaf blade or this leaf is again, like I said, helping in photosynthesis, transpiration, and also the veins are playing a huge role in transporting water throughout the leaf. So now we know what a leaf is. Let's go see what does inside the leaf look like. So if you take a cross section of a leaf, you can see that the leaf on the surface has got a waterproof waxy layer called the cuticle. It is more evidently visible in this diagram. And then just below that layer, you find the epidermis layer. And then below the epidermis layer, we see something called palisade mesophyll cells. Below the palisade mesophyll cells, we see the spongy mesophyll cells. Below that, you find another epidermis layer. And if you see here, this palisade mesophyll cell and spongy mesophyll cells are together called the mesophyll cells. So if you see here, let's go from the below part, the lower epidermis, there's something called stoma or stomata. I think all of us know what a stomata is. Stomata is that part of the plant through which all these diffusion of gases, that is carbon dioxide is being taken in, oxygen is being released out, and water is also being released out to this pore-like structures. It's very tiny structures that you find on the lower side of the leaf. Now again, there are lots of plants which have stomata on the upper lower side and in different ways, but this is just a typical representative, just so that you know what stomata is and how it looks inside a typical leaf. So here, if you see in the lower epidermis, you find something called the stomata. And it is through the stomata that oxygen comes out, carbon dioxide goes in. Now, all of these gases that goes into this leaf, it is having this spongy mesophyll cells for it to circulate within it, or has air spaces that is helping this carbon dioxide and oxygen to be available or to stay here. Now, the palisade cell is something which is what we'll be focusing most on, because it is in the palisade cells that the very important photosynthesis process occurs. So now from what a leaf cross section looks like, we are going to go a bit deeper into these cells called palisade cells, which is where photosynthesis occurs. And palisade cells are, in this case, found on the upper side of the leaf, just below the epidermis. So this is again a very good pictorial representation of the leaf structure. You can see cuticle layer is there. The cuticle layer is a hydrophobic layer that is helping from water loss. Then upper epidermis. Upper epidermis again is helping in reducing water loss. Lower epidermis is the place where you find stomata. Stomata looks like this. When it is open, it will, all the gases will go out or come, go in or come out. And when, in order to optimize how, I mean, if all of the water goes out, then the plant will face water stress, or it shouldn't release all the water, otherwise it will get dried out. So in order to stop that, the stomata will close. Same is the condition carbon dioxide. When too much of carbon dioxide is, in order to restrict the entry of too much of carbon dioxide, stomata will close. Then you see the palisade mesophyll, which is where uh, the photosynthesis process occurs majorly. The spongy mesophyll, where all these gases that enter through the stomata is going to be available. And then again, cuticles. So our main focus now from the entire leaf structure is going to go to palisade mesophyll and stomata. So now let's go a little more deeper into palisade mesophyll. Something I left out to talk about is this vascular bundles called xylem and phloem, which is providing water and nutrients to the leaf. Again, we won't be going too much in detail towards that part of the tissue. So one single palisade mesophyll cell looks like this. It has got a cell wall, it has got a cell membrane, and it has got another organ called organelle, I believe. Vacuole, there is a nucleus, there is cytoplasm, and there is something called the chloroplast. So inside a palisade cell, the photosynthesis is occurring in this organelle called the chloroplast. It is here we find something called the chlorophyll, I think, which is also very familiar with us. Chlorophyll is a pigment that helps a lot in the process of photosynthesis. Chlorophyll is a green color pigment. So again, when we go within a palisade cell, there is something called the chloroplast where the photosynthesis is majorly occurring. So let's now go in, see how a chloroplast looks like. So this is how a chloroplast looks like. A chloroplast is something which has got an outer membrane. It has also got an inner membrane, and within these two membrane, there is an intermembrane space. Within the chloroplast, you can see pancake-like structures or flat sac-like structures. This flat sac-like structure is called a thylakoid. And a bunch of these together is called a granum. Now, all of these thylakoids and thylakoids are embedded in this aqueous media called the stroma. So, our important structures in terms of chloroplast will be this thylakoid and the stroma. So, we see there's an outer membrane, inner membrane, there is an intermembrane space, there is stroma in which we find sac-like structures called thylakoids. A bunch of these thylakoids are called granum, and you can also see from one thylakoid to another thylakoid granum, there is some connectors. This is called stroma lamella. Now, chloroplast, this entire thing is the photosynthetic apparatus of a leaf system. So now we will be going from a chloroplast into thylakoids where very important reactions are happening. Now, you can see chloroplast is not just available in palisade cells. So if you see, we came from a leaf structure into a palisade cell. Inside a palisade cell, we came to a chloroplast. In these chloroplasts are not only found in palisade cells, you can see that they are found in various different places in a plant, especially the spongy mesophyll also consists of chloroplasts. Then this stomata inside the within the stomata there are some cells called the guard cells. You can find chloroplast in there also. And bundle sheath cells, this is another type of plant where there is a different mechanism of photosynthesis, and so on. So chloroplasts and all are not just restricted to palisade mesophyll cells alone, but we will be going in that route just for our understanding, because for us it is not important where all chloroplasts are found. For us, what is important is what is inside a chloroplast. So till now, we went from a plant structure to a leaf to a leaf inside a leaf, from inside a leaf to one of the single tissue inside a leaf, which is a palisade. And from palisade, we went into something called chloroplast. So I I think I'll open up the session towards any doubts or anything that needs to be discussed with respect to whatever I have talked about right now for 5 minutes, and then we'll move ahead to the next part of the lecture. Okay, so if no one has doubts, we'll just move on to the next part of the lecture. So in this part of the lecture, we just now talked about the important parts within a leaf where we are going to be focusing on. Now I'll be explaining to you why we are going in depth into these small parts within a leaf. That is because photosynthesis is occurring in these organs or organelles or cells of the leaf, and we will also be discussing about what photosynthesis and overview of the photosynthesis process. So, like I just said, we went from a plant to inside a leaf to inside one cell to inside one organelle in a cell, if I'm right. I think that's how it's called, right? Okay, so now what we are going to do is go further deeper inside a chloroplast. So inside a chloroplast, now we understood that chloroplast is important. Now inside a chloroplast, our point of focus will be in thylakoid, one thylakoid and the stroma around the thylakoid. So this is just the same thing that I told. Inside a chloroplast, a grana is one that is made up of 10 to 20 sacs of thylakoid. Stroma is where all these are embedded in. And what is a membrane envelope thylakoid system, which is now this thylakoid system is the place where something called the light reaction of photosynthesis occur. So before we go into the details of what a thylakoid is, let us go into what photosynthesis is and what are the different things that are happening. Some basic idea about what the photosynthesis process is. Now, photosynthesis is, as we all know from our school days, CO2 plus H2O plus light will give sugar and oxygen. It looks very simple, although that is not it. The process of photosynthesis is not as simple as this equation. It makes it look like. So, photosynthesis is a process which is broadly divided into two parts. One is the light dependent reaction, The other one is a light independent or dark reaction, or also called the Calvin cycle. So there is a lot of debating as to whether it should be called a independent or dark reaction. So I will be using the name of Calvin cycle to avoid any confusion. So photosynthesis is divided into the light dependent reaction and the Calvin cycle. Now, during the light dependent reaction or the light reaction, what is the input of light and water is made use of to generate outputs of oxygen and two molecules or two substances called the ATP and NADPH. So during the light reaction, as you see in this pictorial representation, water is taken, light is taken, oxygen is released, along with something called ATP and NADPH, that is also being generated during the light reaction. Now this ATP and NADPH that is generated during the light reaction, it is used up by the Calvin cycle along with the carbon dioxide that is taken up by the leaves to produce the sugar, along with two substances called NADP plus and ADP. So if you see here, the water and the light is used in the light reaction to generate two substances called ATP and NADPH, as well as oxygen. And this is used in the Calvin cycle along with carbon dioxide to give us glucose, along with a release of NADP plus and ADP. So again, this is a more detailed representation of this equation where exactly water and light is used, and where exactly carbon dioxide is used, and where is glucose being generated, and which part of the photosynthesis is oxygen being generated. So let us now go a little more deeper into uh, okay, now we know that this is light reaction, this is dark reaction. So light reaction, the goal of light reaction is to use water and light to convert the light energy into chemical energy. Chemical energy is nothing but this chemicals that are produced during the light reaction. This is happening in the thylakoid. Here, as input, light is needed, water is needed. This substances NADP plus and ADP is also needed. Now we get sunlight from sun, of course, water from the roots are providing water, and roots are absorbing water from the soil and providing it. This NADP plus and ADP is being generated from the Calvin cycle, which is being used in the light reaction. Now, in order for this Calvin cycle to happen, they need something called NADPH and ATP. And this is being produced during the light reaction. So these both cycles are very interlinked with each other. Their light reaction is providing the output that is used as an input in Calvin cycle. Calvin cycle is generating an output that is used as an input in light reaction. And this Calvin cycle is occurring in the stroma. So this is why we are going to be focusing on thylakoid and stroma part of the chloroplast. Okay, no, I have moved on. Okay, I'm very sorry. I think I didn't share this. I mean, yes, it's a mistake from my part. I will. Now, are you able to see the slides? Harita, Reshma, yes, yes, it's okay. So I mean, Reshma, can I go ahead with the explanation, or do you want me to explain this thing from the beginning? I'm sorry, I think I didn't share the correct PPT while sharing the screen. So if you guys want me to go through it once again, I can do that. Okay. Reshma says fine. If there's anyone else who wants me to repeat it because this screen wasn't shared properly, please do tell now. Okay, I don't think anyone has a problem. I don't think- Okay. So we see why thylakoid and stroma is important. It is because it is this place where all this light reaction and dark reaction and Calvin cycle is occurring. Thylakoid is the place where light reaction is occurring, and yeah, thylakoid is where light reaction is occurring, and stroma is where the Calvin cycle is occurring. So before we go into light reaction and Calvin cycle, I wanted to introduce this part so that we can understand what is happening within a thylakoid and stroma with more clarity. So now let's go into the thylakoid part of this chloroplast. So now our focus is on thylakoid and stroma. So one thylakoid sac will look like this. It is again a sac-like structure. If you cut through the sac-like structure, you see it has a membrane called the thylakoid membrane. You can also see a variety of substances on this. All of these are complex proteins that are found on this thylakoid membranes. It is these proteins that are helping in the light reaction of photosynthesis. Now, always remember, light reaction is occurring in thylakoid. The Calvin cycle is occurring in the stroma part of the chloroplast. Inside the chloroplast, you have the stroma, which is the medium in which all of these thylakoid is embedded. It is there where the Calvin cycle occurs. It is in the thylakoid where the light reaction occurs. So you see this thylakoid membrane is enclosing something called the lumen. Lumen is again an aqueous medium where which is found inside within the thylakoid membrane. And on the thylakoid membrane, you can see something named 1, you can see something named 2. There's this orange thing that looks like a mushroom, and again something like this. So all of these are complex proteins. Now, these let's go into these complex proteins one by one. So here what you see here is named as 1 is called something called photosystem 1. What you see here is 2 is called photosystem 2. This is called cytochrome B6F, and this orange thing is called the ATP synthase. So as far as we are concerned, four important proteins that we need to focus on is photosystem 1 and 2, ATP synthase, and cytochrome B6F also not very important, but then yes, these are four important uh, protein complexes, very complex proteins made up of different small protein particles. So you see all of these are on the membrane of the thylakoid. So it is basically in the membrane of the thylakoid, the membrane of the thylakoid, that is a very important center for the light reaction, uh, occurrence of light reaction. So what is this light reaction? So this is again the same things that you had seen in here. This is the photosystem 2, this is cytochrome B6F, this is photosystem 1, and this is ATP synthase. Now, if you see here, the first protein complex that is taking part, or that is starting this light dependent reaction, is the photosystem 2. Photosystem 1 and 2 are the two uh, protein complexes that has got this chlorophyll pigments in it. This is the chlorophyll that we all have learned about, but it is found in the photosystem 1 and 2 of the thylakoid membrane. So in the photosystem 2, what happens is there is something called, there is an oxidation center in this oxygen evolving complex in this photosystem 2 that helps in splitting the water that is taken up by that is provided to the leaves of the plants into oxygen, protons, and electrons. So water is split in PS2 to give to release oxygen, protons, and electrons. And this is the only place in the entire biological system where natural oxidation of water is happening, splitting of water is happening. And this process is called photolysis, that is splitting of water with the help of light. Now, when this splitting of water is occurring, like I said, protons are released, electrons are also released. So, protons are these H plus ions. Electrons, which are released into this PS2, these electrons will be in a very low energy state. So, what happens is this chlorophyll molecule in PS2, it absorbs light, it absorbs photons from the light that is available and excites this electron. This excitation of the electron will result in the increase in the energy. Low energy electrons are going to have a spike in the energy of the electron, and this will help in this electron being transported across this membrane. It will get transported to here, and then again it will get transported to here, and then finally again it will reach PS1. So after this water molecule is split, the electrons that are available here gets into a high energy state with the help of P680, which is the chlorophyll molecule, which takes in light to help in the increasing the energy of this electron, which is eventually transported across all of these protein complexes to reach PS1. So as this electron gets transported from PS2 to PS1, the energy of this electron is going to go down and go from high energy state to a low energy state. So what happens when this energy is going from high energy state to a low energy state is one energy gradient is created. Now, what is the purpose of creating this energy gradient is it helps in these protons that are there in stroma. So the stroma has a lot of protons already. So and lumen also has a lot of protons because the water splitting is going to increase the concentration of protons in this lumen. So what happens is the protons in the stroma, for it to come to this high concentration, it cannot on its own because the concentration, because of the concentration of H plus ions being less in the stroma and more in this lumen, it needs some push to come to travel from here to here. So this electron transport is going to create an energy gradient that helps transporting the H plus ions in the stroma to the lumen. So this is the purpose of electron transport chain between PS2 to PS1. It is to create an energy gradient for this H plus ions from stroma to travel to lumen. Now, what is the requirement of this H plus ion to come from stroma to lumen is that once this concentration of H plus is increased as a result of all these H plus ions coming here, plus all of the H plus ion that is obtained from the splitting of water, this concentration of H plus is something that will drive this protein complex called ATP synthase. Now, if you had listened to the earlier part, we talked about something called ATP and NADPH, which is being generated, which are the outputs during light reaction. So it is this ATP synthase with the help of this concentrated H plus that will drive this ATP synthase to rotate like a motor. And during the rotation of it, it will take H plus ions from the lumen back into the stroma. Now, what is the necessity of this ATP synthase to take this H plus ions from the lumen to the stroma is it is this H plus ion which will help in ADP which is available in the stroma to attach with an inorganic phosphate to become this ATP. So ATP, the output that we are seeing in a light reaction, this light reaction is formed as a result of jamming of this ADP and an inorganic phosphate with the help of H plus ion that is released by this, that is taken from this lumen to the stroma by this ATP synthase. So now we know how light and water is being used. Now we know how light is being used, and now we know how we got this output ATP. Now, Coming back to PS1, when this electron is transported from PS2 to PS1, as it reaches here, this one's energy also will go down. So in order to increase the energy of this one, again the chlorophyll molecule that is present in PS1. You see the name of the chlorophyll molecule here is P680. Here it is P700. This P700 chlorophyll molecule will again absorb the photons from the light and excite this electron to a high energy state. Now, what is this electron going to do is it is not going to take it is also getting used in an electron transport chain, except that this electron in will travel through these proteins called ferredoxin and ferredoxin NADP reductase and use the H plus in the stroma to reduce this molecule called NADP plus. It is NADP plus is the input. So it will reduce, these electrons will provide energy for this H plus to reduce NADP plus into something called NADPH. So this is how we are getting this output of the light dependent reaction. So we saw that how sunlight is being used. We saw how water is being used in this place. We saw NADP plus, which is an input, which is basically there already in the stroma, is being used to obtain NADPH. We also saw that how these H plus ions and ATP synthase is going to help in jamming this ADP and PI together to form ATP, which is another output of the light reaction. So again, you can see this is a representation of the electron transport chain. How water is providing electrons into this P680. P680 will take up photons from the light and the energy will get increased. This electron will get transported across all these proteins and then reach PS photosystem 1. Again, it will get excited, which will these electrons will be used in the production of NADPH. Along the course of this, the concentration gradient increasing over here is going to drive the ATP synthase to produce ATP. So, those of you who are new to this, I hope this part is clear. Maybe just go through this diagram for a minute or two, and then you can post in your doubts now. So how many of you are not familiar with anything related to plants and photosynthesis? Can you raise your hands using that? There's some raise hand option over here. Everyone seems to be knowing, so it's good. Okay, so if you don't have any doubts, you can just post, you can just reply in the chat, and I'll continue with the class. Okay, I don't seem to be getting many responses, so I think I'll assume that nobody's got any doubt here. Okay, no doubts for now. So I'll continue. So right now we were looking at the light dependent reaction. So just a summary, photolysis of water is happening. It is generating protons, electrons, and oxygen in PS2. The electrons from water splitting at low energy is excited by the light that is absorbed by chlorophyll molecule P680. This high energy electron is transported across the membrane from PS2 to PS1. The electrons lose its energy as it reaches the PS1. Then the chlorophyll molecule in PS1 excites the electron, which increases the energy of the electron that helps in the process of reducing NADP plus to NADPH. During the course of it, again, high energy electrons is facilitating the energy gradient that is resulting from the electron transport is helping in the movement of H plus ion from the stroma to the lumen. This increases the concentration of H plus in the lumen, which drives the ATP synthase, pushing H plus ion out back into the stroma, which is again initiating that ATP production by the ATP synthase. So what we looked at is the light reaction. Now, the next part of it is the Calvin cycle. So Calvin cycle is happening in the stroma. So this is the thylakoid. This Calvin cycle is occurring in the stroma. Now, as you can see here, the goal of the Calvin cycle is to use the chemical energy. We know the output of light reaction is NADPH and ATP. Those are chemicals. So that chemical energy is used to fix carbon dioxide to create a product that is converted into sugar, which is necessary for the plants. So till now we saw where water and light is used and how oxygen is being released. Now what we are going to see is how carbon dioxide is used and sugar is released. So this process is occurring in the Calvin cycle, and Calvin cycle is making use of carbon dioxide, the NADPH that is produced during the light reaction, and the ATP that is produced during the light reaction is going to be used in the Calvin cycle. So let us just go a little bit deeper into the Calvin cycle. So in the Calvin cycle, it is divided into three stages. One is the carbon fixation. The carbon fixation is nothing but the reaction of a molecule called RUBP. RUBP is ribulose by 1,5-biphosphate. This RUBP molecule will combine with carbon dioxide that is taken in by the stomata, and with the help of an enzyme called Rubisco, this two will react together to form this molecule called 3-PGA. Now, if you see, this has got 5 carbons in it and 3 molecules of it. So, 3 molecules of carbon dioxide will react with RUBP with the help of this very important enzyme called Rubisco and form something called 3-PGA. This stage is called the carbon fixation or carboxylation. That is combining of carbon dioxide with RUBP with the help of an enzyme called Rubisco. Now this 3-PGA formed, you see, has a negative sign here. So what happens is this 3-PGA with the help of this ATP that was generated during the light reaction will get, and NADPH will get converted to, will get converted to a reduced form. With ATP, it will get converted to a reduced form of 3-PGA. You see this negative sign is going and it is being something called CHO. Then with the help of NADPH, it is getting converted into another molecule called G3P or GA3P. You can use it both ways, I believe, because in some places they have given GA3P, some places they have given G3P. So this 3-PGA molecule with the help of ATP and NADPH is getting converted into G3P, that to 6 molecules of G3P. Now, why are we giving so much importance to the number of molecules is that during this reduction of PGA, we see that ATP is used, when ATP is being used, that is the chemical energy of ATP is used, it gets converted back to ADP, and when the chemical energy of NADPH is used, it gets converted back to NADPH. Now, this is how in the light reaction you find this NADPH and ADP already available in the stroma. The Calvin cycle is what is providing these molecules for the synthesis of ATP and NADPH during light reaction. So we found out how there is NADPH and ADP existing in the stroma already. It is because of this Calvin cycle. So without Calvin cycle, there can't be light reaction. Without light reaction, there can't be Calvin cycle. So it is a it is a process. These are two processes that are providing and taking from each other. Now, after the reduction of the 3 PGA, 6 molecules of 3 PGA to 6 molecules of GA3P, what happens is one molecule of GA3P goes in the manufacture of that most important thing that the plants are trying to manufacture, which is carbohydrates or sugar. So out of 6 molecules, one molecule go for sugar, and the remaining 5 molecules are again taken up for regenerating this RUBP molecule. So during the Calvin cycle, most of the energy is used in keeping the cycle going on rather than the carbohydrate generation. So this is a very important cycle with respect to the fact that the energy has to be optimized, because most of the energy over here is going to generate this 5 molecules of GA3P that is going to be used for regenerating RUBP, which has to act with carbon dioxide, and only one molecule is going for the sugar production. So it is very important that the energy centers, which is ATP, NADPH, all of these are the energy providers for this reactions in this Calvin cycle to happen. So it is very important that we make use of all the plant or wherever it is occurring, makes use of this energy in a very optimal manner to generate the for the purpose of generating sugar. So this is what is called the Calvin cycle. So we'll be going more into detail into the Calvin cycle in the next class. As of now, what we have to keep in mind, some very important terminologies that you have to keep in mind is RUBP, Rubisco. This reaction is called carboxylation. That is the RUBP plus carbon dioxide reaction is the carboxylation. And during this process, there is the sugar molecule. There is a molecule that is helping in the manufacture of sugar. Also, the molecule not only help in the manufacture of sugar, but it also help in the regeneration of RUBP. So this regeneration of RUBP is an important thing that you need to have in mind. Carboxylation is an important thing that you should have in mind. Then Rubisco enzyme is an important thing that you have to remember. RUBP is an important thing that you have to remember. ATP, ADP, NADPH, NAD plus. These are all important things that you have to remember. We don't need to know all the chemical formulas or chemical structures of these molecules. Neither do we have to remember the names of these, because all these are I'm just explaining so that we can have a clear understanding before we go into the plant modeling. So these are some of the abbreviations that are used in the lecture. What is RUBP, Rubisco, what is PGA? In case you want to go more deeper into it, you can use this table, what is being used in this study. So our lecture will be winding up with this much portion. Next class, I'll be going a little more deeper into Calvin cycle and the photosynthetic pathways. So I would suggest everyone to go through the BioCro software that I've shared. I'll be sharing this PPT as well as the lecture. So please go through the software. And those of you who are not familiar with these concepts, go through the slides or read more using your own resources. These are the stuffs that will be required as a foundation for the plant modeling course. I also have shared a Google form where you can put in your doubts later on when you're studying it on your own. Apart from that, if you have any doubts now, you please feel free to ask. I think I'll be here for 20 minutes more. So do ask anything if you want. And if you don't have any doubts, you're free to leave the session. Thank you for attending the session. I'll be updating more in the WhatsApp group and through mail. Thank you for Vishwa. Manish, if you don't have any doubts, you can exit the session. Hello, Mayank. Do you have any doubt? Harita, you can exit the session, no problem. Thank you for attending.